Also, you don't want to have an enzyme active when there's nothing for it to do. That's not going to work either. And you don't want an enzyme, for example, to make too much product because that's going to throw all the balance off. For example, you have an enzyme that, let's say, breaks down glycogen into glucose. If you need more glucose in your cells and you need more blood glucose, then you're going to turn that enzyme on. But at some point, it's going to get to a point where the glucose level starts to get too high. You want to shut the enzyme down. So we want to be able to turn enzymes on and off as needed to maintain homeostasis. So the way that we regulate enzymes is through what are called inhibitors. An inhibitor is something that has the ability to shut an enzyme down. Some toxins are enzyme inhibitors. The reason they make you sick is because they shut down particular enzymes. However, your body makes these naturally as well. So, first, competitive inhibitors. Competitive inhibitors are those that look very much like the substrate, but if the enzyme binds to one of these inhibitors, basically nothing happens. It can't break it down. So it's sort of like if I was supposed to run around the room and let's just say shake hands with only boys, but I had some girls in the room too. Every time I bump into a girl, it's going to slow me down because I'm not supposed to. That would be like a competitive inhibitor. They look similar to a boy. They still have, a hand. They could shake my hand, but they're not the actual proper thing I'm supposed to be shaking hands with. The rule about competitive inhibitors, and it's a guaranteed thing you would be asked on a test, is that if you increase the amount of substrate, you can overcome it. The inhibitor won't have a lot of effect. For example, again, if I'm only supposed to shake the hands of boys, and in the room I have 10 boys and 10 girls, my progress may be kind of slow because I have sort of a 50-50 chance of bumping into the right thing. But let's say that, again, I'm supposed to shake hands with only boys, and there's still 10 girls in the room, but now I add 70 boys. Now, those 10 girls are probably not going to affect things that much because my chances of bumping into the correct thing, a boy, are going to be much higher. I used to have everybody do in this lab a second part where, because you may have noticed there's some pink papers here, where I had you add pink papers to the bag. And the idea was that if you reached into the bag and you picked up a pink paper, it was like a competitive inhibitor. You can't actually rip the pink paper. It feels just like the white paper. It fits the enzyme just like the white paper. But every time you pick up a pink paper, you can't rip it, so you drop it back in. Imagine how it's going to slow you down. But if I only had one pink paper in the bag and I added 100 more white papers, that pink paper is probably not going to make much of a difference at all because the chance of me actually bumping into one is going to be very low. So, competitive inhibitors, and I'm going to show you this in graph form in a second, can be overcome by adding more substrate. But they do slow enzymes down. The other kind of inhibitor is called a non-competitive inhibitor. I've also seen the term sometimes where they're called allosteric inhibitors. Allosteric or non-competitive inhibitors do not bind or compete for the active site of the enzyme. What they do instead is they bind to an alternative area on the enzyme, and it changes the shape of the active site. In other words, if we imagine my enzyme as little Pac-Man here, and this is the other site I'm talking about. We're going to call it sometimes called an allosteric site. This is my active site. My substrate looks like this, and my substrate normally fits in that active site, and then they react. When the non-competitive inhibitor binds, it binds to my enzyme in this other site. Notice what happens. Since enzymes are proteins, now the active site of my enzyme has changed. The substrate is not going to bind to it anymore because there is no active site. Because by the enzyme binding to this inhibitor, it molds into a different shape and it no longer fits the substrate. If I add more substrate, this is not going to speed things back up because every enzyme that's bound to the inhibitor is basically out of the game. In other words, again, if I added more girls to the room and I'm shaking hands with, supposed to shake hands with boys, I add more boys, it'll speed things back up again. But imagine that instead, what I did was all of you guys were enzymes and you're, let's say, picking papers out of the bag. Competitive inhibitor, I add some pink papers to the bag. It's going to slow you down, but the more white papers there are, the less effect it would have. Let's say instead I choose 10 of you and I tie your hands together. This would be like an allosteric or a non-competitive inhibitor. Those people whose hands are tied together are out of the game. They don't have an active site anymore. So the rate is not going to speed up. Adding more white papers to the bag isn't going to make things work any faster. If you have now less enzymes available to do the job. So competitive inhibitors compete for the active site on the enzyme. Non-competitive inhibitors bind to an alternative location on the enzyme and change the shape of the active site so that enzyme is shut down. Again, you naturally make these to control reactions, but also some poisons, toxins, antibiotics on bacteria, pesticides, the way a lot of them work to kill weeds or to a neurotoxin or whatever it may be, a lot of them, the way they work is they're inhibitors. So they inhibit an enzyme, which means the cell can't do its job, whatever the job that enzyme was, or job of that enzyme was, and that cell may die from it. But again, I don't want you to think that inhibitors are always bad things. Your body makes inhibitors on purpose because you don't want to make too much of certain products. You need to shut enzymes down sometimes before they do too much work and make too much of something, and now that's throwing the balance off. This is a graph. You should understand this graph. Remember, this is the graph where I increase under here, the S with the little brackets means concentration of substrate. Here's my regular enzyme. Remember, do you remember what we called this when it got to this point? It's max, where all the enzymes were working. Do you remember what that was called? So that was called being saturated. Every enzyme's working at maximum speed. Here's what happens if I add a competitive inhibitor. See this one right here. At low concentrations of substrate, the competitive inhibitor slows things down, but the more substrate I have, basically the competitive inhibitor has very little effect. And then here is a non-competitive inhibitor. With the non-competitive inhibitor, every enzyme that the non-competitive inhibitor binds to is basically removed. It can't do any work anymore. So now you basically have half of the enzymes working. Because typically you don't add enough inhibitor that every enzyme is shut down. But if you add, whatever inhibitor is added, it usually shuts down some of the enzymes. So those enzymes that are shut down, it doesn't matter if I add more substrate. The enzymes, there's less factory workers. So you're not going to be able to speed the reaction back up because those enzymes are out of the game. You have less enzymes actually working. So that's a graph that kind of shows you what the difference is between competitive and non-competitive. Here's a little picture too. Competitive, the substrate and the inhibitor compete for the active site. Obviously, every time it binds to an inhibitor, it's going to slow it down. All right, and then non-competitive here. So the non-competitive binds to another area on the enzyme, changing the shape of the active site. Every enzyme that gets bound with a non-competitive inhibitor is no longer able to participate in the reaction, so that's going to slow the reaction down. So that's the difference between the two. All right, and that brings us to the last thing here, with way that we control enzymes, which is what's called negative feedback or feedback inhibition. Remember how a lot of pathways we talked about at the very beginning of the lecture are what are called metabolic pathways. It's not one reaction, but it's a whole bunch of reactions. So here's my original substrate that comes in, and you have several enzymes. Enzyme A converts it to B. Enzyme B converts it to C. And this is the final product that your cell needs. So it's sort of like an assembly line. What can actually happen is like the enzyme is actually self-regulated. This product is being made because it's something that the cell needs for a job. But this pathway, this assembly line is going to keep going until something tells it to shut off. So if you keep making product, you might eventually get to a point where you have too much of it. So what will actually happen is kind of beautiful. Your cell is set up so that the product is actually an inhibitor. It will actually go back here, and it actually fits on enzyme A. So this product can actually go back, hook to enzyme A, shut it down, and now the whole assembly line stops until you use the product up. So rather than having to have some separate signal that controls it, it controls itself. So you start, let's say, breaking down glycogen to make sugar. The sugar levels maybe are starting to get too high, but sugar might be able to go back and shut it down. Just like if you guys were an assembly line putting stuff together and the person at the end starts to get overwhelmed. They could, send somebody over and say, hey, shut it down for a couple minutes, we need to catch up. Basically, the product is what does that. As the product gets used up, this inhibitor will get used because it's a product, and that'll turn everything back on again. I actually have another picture on the next slide, I think of a more realistic one. So this is the active site, if we start up here. The isoleucine is the product. So this enzyme breaks threonine. Again, the names of these things are not important. So threonine is the original reactant, the substrate. This enzyme binds to threonine, turns it into these intermediates, several enzymes. The final product that the cell needs is isoleucine, which is, it's an amino acid, but again, the names are not important. Isoleucine actually binds to this original enzyme and shuts it down. So as the cell makes isoleucine, if it starts to make too much, the isoleucine itself will shut the pathway down. As the cell uses up the isoleucine, the enzyme will go back to its correct shape, and the pathway will get turned back on again. So this maintains homeostasis. These pathways can be self-regulated. The products actually shut the pathway down until they're used up, and everything will just run without any external signals even being necessary. And that brings us to the last thing here. So the last thing is cofactors and coenzymes. Some enzymes do not have the correct shape when they're originally manufactured. They need a helper. Cofactors and coenzymes are helpers that give enzymes their correct shape. The only difference between them is that cofactors are inorganic and coenzymes are organic. So cofactors are things like zinc, iron, iodine. You know, the things on a bottle of vitamins that are minerals, that are ions, those are typically cofactors. And coenzymes are vitamins, like vitamin A, vitamin C, vitamin B12. All of these things, what they really are, when you take a multivitamin, you know, they tell you, oh, take a multivitamin because your body needs it. What vitamins and minerals really are in that bottle of vitamins are cofactors and coenzymes. Some enzymes don't have the right active site shape without a cofactor or coenzyme bound to them. So when you have a vitamin deficiency, that's why you actually end up with scurvy or beriberi or rickets or whatever the name of the disease associated with that vitamin deficiency is. Believe it or not, vitamin A helps you see well at night. It's an enzyme, it's a coenzyme that helps an enzyme in your eyes function under low light. So if you have a vitamin A deficiency, you can actually have what's called night blindness because the enzymes in your eyes won't work properly. They won't be stimulated under low light and you can't see at night. And all you got to do is increase your vitamin A, and you'll suddenly be able to see better at night. Doesn't mean everybody will have, you know, X-ray vision if you take lots of vitamin A. But bottom line is, vitamins and minerals, cofactors and coenzymes, not every enzyme needs one, but some enzymes require them. They hook to an enzyme and help it have the correct conformation. And so that's why a lot of those things are necessary. Most people get what they need in a normal diet, but that is what a multivitamin, that's why you may need a multivitamin. So when you're wondering, you know, well, what are vitamins really? That's actually what they are. Cofactors and coenzymes that hook to specific enzymes in your body that need them and help them to work better. So the enzyme takes on a better shape when the cofactor or coenzyme is there. And that's actually the end of the chapter. That's everything. It's a lot of stuff.